Welcome back in this session, that is session number 4, chapter 1, lesson 4. We will be looking at biologists, tools, and technology. We will be looking at biologists, tools, and technology. So technology continually changes the way biologists work. Just like any other fields of scientific fields or even social fields, with emerging technologies, things are changing day in, day out. This means that even in biology, as continually things are changing, the people also have to change with time. That is what we are going to look at. In this instance, it means that as new inventions come up, then the people have to cope with the new type of life. Otherwise, if they don't cope with the new type of life, then we will not have any development in terms of biological studies. The tools that are there are many, but the main ideas that we will be looking at in this unit will include imaging technologies, provide new views of life, complex systems are modeled on computers, and the last part is that the tools of molecular genetics give rise to new biological studies. We will start with the first tool, which is very important, and the tool which in fact we can say brought a breakthrough in science. This tool is nothing other than the microscope. Imagine biology without a microscope. If, for example, we don't have a microscope, then we will not be able to see microorganisms. If we can't see microorganisms, then it means that we will not know how diseases come about, how diseases come about, or what are the reasons of diseases. We will not know this. So technology continually changes the way biology works. Technology continually changes the way biology works. Technology also plays a major part in rapid increase of biological knowledge. It also plays a major part. It also plays a major part. It also plays a major part in the rapid increase. In the rapid increase. So in the rapid increase of biological knowledge. With the emergence of microscope, many breakthroughs have come up in terms of development of biology. Until the late 1600s, until the late 1600s, no one knew about a cell, knew about a single cell. No one knew about a single celled organism. For example, celled organism. But as inventions came up, or with the invention of the microscope, the first microscope, microscope helps to magnify objects that could not be seen with our naked eyes. Microscope had a combination of lenses that provide clear images. We have different types of microscopes, and we will look at them one by one. We will start with the first type of microscope, which of course is nothing but the light microscope. The light microscope. We have different types of microscope. We have what we regard as being a light microscope. The diagram you see here is of a light microscope. A microscope has different parts, as we will be looking through. The first part here, these are the objective lenses. These are the objective lenses. We have those ones which have higher magnification, and we have those ones which have low magnification. That one with high magnification is regarded to be a high power. So we have high power and low power. So low power and high power. High power magnifies more than the low power objective. So apart from we have even the middle ones, but normally in our biology class we normally use the low power and the high power. So the next thing that we will see is so as we look at we will see that we have a slide which is put on the stage. This portion is the stage. You can rotate the objective lens so that you select the right objective lens that you want to use. As you observe, you will see you will see, of course, things that are magnified. In this case, the microorganisms. If we take an example, you go into the jungle or the forest or in a pond, you collect a drop of water, or in this dog's mouth, it is having billions of bacteria. How can we get this? You get just a drop of the liquid, you put it on a slide, then you can cover it. Or we want to see if someone has a wound like this one, if he has any infection of bacteria or viruses, then we will take the sample and we will put on the slide and observe. Or even we will see very many things here as we use the microscope. The microscope that we have here is a light microscope. If you look at it carefully, you will see that microscope will make things look wide or magnify them. Microscopes will magnify things. Here we have the stage, we have source of light, we have the eyepiece, which of course also has a lens. So together, they work together. We have had many developments in microscope. Earlier microscopes were very simple microscopes, but now we have more complex microscopes. Even now we have the development of even the electron microscope. We will discuss the electron microscopes one by one. What you see here is just a sample of what has been observed. So we will push this a little bit so that we look at other types of microscope. What you saw there was just but a light microscope. That was a light microscope. So that was a light microscope, like the one that we use in schools. Then we have electron microscopes. Electron microscopes. With the development of electron microscope, then biology even became more advanced. With the development of electron microscope, biology even became more advanced. With the development of electron microscope, biology became advanced. And we are saying here that we have two types of electron microscope. We have one, the scanning electron microscope. One, we have the scanning electron microscope. So, scanning electron microscope. And two, so this is one, and two, we have the transmission electron microscope. In our discussion, we might be using abbreviations as SEM to mean the scanning electron microscope, or we may use the TEM to mean the transmission electron microscope. We will begin with the first one, which is the scan electron microscope. So usually a specimen is coated with a very thin layer of metal. In this case, if you are using this microscope, then what will happen is that the specimen is normally observed through a beam of electrons. So usually the specimen's surface. So the specimen surface is coated. The specimen surface is coated with a thin microscope. With a thin metal. With a thin layer of metal. After that, what will happen next is that this metal deflects the electrons. So as you try this metal, what it does, it deflects the electrons. When it deflects the electrons, this of course is either observed as a three-dimensional image from a computer or even from the microscope itself. So this will give you 3D or three-dimensional image. This will give you a three-dimensional image, which means it is more clearer in terms of clarity. The scan microscope, on the other hand, the transmission electron microscope, on the other hand, what happens with it? So transmission electron microscope works in this case like this. A thin specimen is placed on the microscope. A thin specimen A thin specimen is placed. A thin specimen is placed on a slide. The next step, in this fact, transmission electrons are transmitted through this slice of specimen. Then electrons are transmitted through. They are transmitted through this thin slice. Because electrons are transmitted through this, and even in the scan electron microscope, together with the transmission electron microscope, we cannot have a living object. We cannot have a living object or a living thing observed. So we can't have a living organism observed here. That is the disadvantage of electron microscope. Even though it gives high, we have the electron microscope. We have seen that we have two types, and then we have the light microscope. The light microscope provides the advantage in that you can see live specimens. Here, you cannot see live specimens. In this case, you cannot see live specimens. So the specimen is dead. So you can't see live specimen here. The light microscope is also cheap. The light microscope is cheaper. It's cheaper and easier to use. The electron microscope is more expensive. Light microscope can be used anywhere. So long as there is a source of light. Even if we don't have electricity, we could still use a light microscope. But the electron microscope is more dependent on electricity. So here, you must have a source of electricity. A source of electricity. We will go to the next portion here. We will stop here for now. Then in the next session, we will be looking at images which come up in these, as you observe these materials. You